So, good morning, everybody, and welcome to the Code of the Month, number 16. We are very happy today to host the MultiXScale COE. We will see a presentation given by Jean-Noël Grat and Rudolf Weber from the University of Stuttgart about the code ESPResSo, which is a code combining different methods for simulating materials. So, Jean-Noël and Rudolf, the floor is yours.
Thank you very much for having us, and thank you everyone for coming. So we would like to talk about the simulation package ESPResSo, which has a focus on the physics of soft matter systems and also of systems in process engineering in terms of the method that it provides. For these kinds of simulations, for these kinds of research questions, we typically need a combination of particle-based and of continuum-based approaches to describe the systems. So examples that you can see on the slides would be a coarse-grained model of a polymer, which is translocated through a nanopore. That is, for instance, the technique which is used for DNA sequencing. Coarse-grained here means that we don't resolve individual atoms, but we zoom out further and basically use a more coarse resolution in the description. And the second picture is of an ink droplet advecting in water. So we have diffusion and advection going on here at the same time. That is an example of like the continuum methods that would be present in the software. Let's move on. To give you a bit of background for the applications or research questions for which the software is used, let me very briefly talk about soft matter science. So soft matter science typically happens at a scale between nanometers and microns. And at this scale, the energies that are required to influence the system, for instance, to deform it or to move components, are roughly comparable to the thermal energy. And this is why Brownian motion is crucial in these systems. So nearly always this randomness provided by Brownian motion is present in these systems and has to be accounted for in the simulations. Here you see a video of a pollen undergoing Brownian motion in a microscope. So moving on. The energies are comparable to the thermal energy. So Brownian motion is important. At the same time, nearly all of soft matter science, like for instance, a red blood cell in the bloodstream, would be in the presence of a solvent. So to understand what soft matter systems are doing, it is typically also necessary to include the dynamics of the solvent. Now, the embedded objects like a red blood cell or a pollen or whatever is much bigger than the water molecules. So we would not need to know where each water molecule is. Instead, for the solvent, we would use a continuum description. But solvent dynamics is often crucial. Moving on. So, like taking this together, often a good modeling approach to soft matter systems is to describe them as interacting particles which are embedded in a solvent. And both the particles and the solvent are moving, and they can also influence each other. So examples would be a polymer chain like the DNA shown here, but also the nanoparticles in a ferrofluid, which is basically a fluid that contains magnetic nanoparticles. So here again, the coarse-grained description, there is no point knowing where every atom of these magnetic particles is. We zoom out and take one of these nanoparticles as a particle in our simulation model. Moving on. So in terms of the simulation methods that we have to describe these combined particle and continuum models, we would have molecular dynamics, primarily for the dynamics of the particles. So that is basically integration of Newton's equation of motion for interacting particles with some extras, for instance, to manage temperature. There's also Monte Carlo methods, that is methods which introduce some randomness in the system. We use this, for instance, to model chemical reactions between particles. Lastly, there's the continuum techniques. So for the solvent dynamics, lattice-Boltzmann hydrodynamics is a very useful approach for this length scale of nanometers to microns. And for immersed solutes, like the video with the ink drop that you saw in the beginning, we use a solver for diffusion, advection, and reaction equation. So these are the basic algorithms that ESPResSo provides in like conjunction with specific features for individual applications or research questions. And I would now pass over to Jean-Noël, who will present some of ESPResSo's capabilities by showing you some examples.
Thanks. So the first application we would like to display is about energy materials. If I can get the slide to move. So in this particular example, we have something called a supercapacitor, which is made of these red and blue particles, which represent a room temperature ionic liquid. So each sphere represents an entire molecule. Again, this is a coarse-grained model. And the black mesh that you see on left and right are the porous electrodes of the energy material. Now, the two ions in the liquids are mixed in the bulk solution, but as you apply energy difference between both electrodes, you will see selective diffusion of the ions into each electrode. So one will be more or less negatively charged, and the other one will be more or less positively charged. And these so-called supercapacitors have many advantages over regular electrolytic capacitors. One of them is durability. They will allow you to cycle through charge and discharge for a much longer period than regular capacitors, but also they can charge a very large amount of ions, and consequently you can discharge them very fast. So in their use, they are slightly different because of the charging and discharging dynamics. So they have different applications. One of them is in electric vehicles. You can have the lithium-ion battery to power your engine, but if you want to accelerate very quickly, having next to it a supercapacitor is very interesting because supercaps can discharge their power very fast, something that a lithium-ion battery cannot do easily for safety reasons. To calculate the interactions between these charged particles, we need specific numerical methods, which depend on the geometry of the system. It depends on whether you have a 3D periodic system, or in this case, a 2D periodic system. And if you want to model curved surfaces for the electrodes, you might need extra algorithms to deal with the electric contrasts. This is of particular interest for MultiXScale because one of the scientific goals is to model a supercapacitor on a very large supercomputer. But it's so interesting for us because charged soft matter is ubiquitous. Another example is biopolymers. They almost always have a charge, and it's important to have numerical methods to properly account for these interactions. In this particular example, we have a DNA polymer, which is moving from one reservoir to the other through a glass nanopore under the influence of an applied electric field. And the small spheres you see in both reservoirs and inside of the nanopore present dissolved salt ions. Now, when you have a polymer enter the nanopore, you will partially block it. And this will make chloride and sodium ions much less likely to go through the nanopore. And you can actually measure the current of the salt as a function of time. And when the DNA enters the spore, you can see a small deviation in the current. If you have a very sensitive device, you can actually see a different deviation depending on the base pair on your DNA polymer. And now you have a technique to actually sequence a DNA molecule. If we go now into the micrometer range, here is an example of a system where we have red blood cells which are moving through some constructed geometries. this is a simulation where a channel was created with different sizes in both branches, such that small objects like red blood cells can easily go into one branch, but the white blood cells in yellow cannot. And you can already think about applications in medical devices to separate white blood cells from other elements of the blood and carry out tests on different components. And to achieve this, we have different techniques. The one in this particular study was using a mesh to describe the white blood cell with around 300 vertices. And on each vertex, we introduced a small deformable particle. And we use different types of bond interactions to make sure that the object, the blood cell itself, is soft and deformable, so that it can actually conform itself and squeeze into these tight geometries. But as soon as it leaves the tight geometry, it can relax to its default shape. If we look at even larger systems, it might become interesting to not describe every single particle as a sphere, but rather as a concentration on a grid. So this is a way to simulate very large systems in a very efficient manner. And you can see on this picture, we have 3 different snapshots of the same system, but each snapshot shows you the concentration of a different species. And you can clearly see that the resolution is so small that you can see the outline of the solid object in gray, and you can see the individual grid points. In this particular simulation, we were interested in the chemical reaction between 2 species called EDUCT 1 and 2, as they are injected into a system with a free flow such that you observe the onset of turbulence. If I play the animation, you can see this wave-like pattern. And as the 2 different species slightly mix and overlap, they will chemically react and form the product. It's a bit difficult to see because the chemical reaction is very slow, but towards the end of the channel at distance 80, you can see 3 new sine waves in the product, which correspond to the newly formed chemical. We can also carry out chemical reactions with particle-based systems. Here we provide multiple methods. I will not go through the theory behind them in the sake of time, but I can show you very quickly the 3 different applications. The first one on the left is a paper about a polymer star, which has chemical groups on them which have a charge. But these are acidic or basic groups, which means if you introduce an acid in the system or an alkali, for example, through titration, you can actually convert the groups from an acid to the conjugated base. And as you do so, you can actually remove the charge on the polymer surface and cause the polymer to collapse due to hydrophobic interactions. So in this case, we have to simulate a system where the number of particles over time is changing. Not only that, but we are actually adding a lot of new particles to carry out the chemical titration. In the middle picture, we have a 3D network of a polymer hydrogel, which has this characteristic that when you add moisture to the dry powder, it will absorb it. It will release protons in a solution because all the acidic groups want to be negatively charged, and the polymer will then be extrostatically repelled by itself, and it will swell. But if you add salt to the system, you will screen the Coulomb interaction, and the polymer will now collapse. And so you will observe the swelling behavior of the network. On the third image on the right, we have a slab on which we grafted polymer chains, which are able to chemically interact with metallic ions, which can have a charge of +1 or +2 electric charges. And in this case, when we introduce metallic ions, we have +2 charge, the polymer will actually form a coil or a loop to maximize interaction with the salt. And so we can observe different types of swelling behavior depending on which ions are present in the system. We can also steer the simulation with Python. In fact, most of the simulation using ESPResSo use Python in one way or another, which allows us to connect to many of the numerical simulation packages available in the Python ecosystem. And this gives you great flexibility in how you steer the dynamics of the system. In this particular example, we're looking at the formation of a biofilm from bacteria. So we start with one bacteria, which attaches itself to some solid surface. It will absorb nutrients in the liquid, and it will start to grow, more or less exponentially, until it forms a biofilm and fill the entire porous structure. This is of interest in different areas. For example, you can make concrete mix with small pellets containing this bacteria. And as you cast your concrete structure, it can be a bridge, a building, or any other structure, the pellets of microbes will be stuck in the solid object. And after 20, 30 years, when the concrete starts to age and cracks from the surface, moisture can get in, but these pellets containing these bacteria will actually break, and now the bacteria can absorb the moisture, the carbon dioxide in the air, as well as the calcium hydroxide in the concrete, and it will start to metabolize all of these chemicals and produce calcium carbonate, which will precipitate in the form of limestone. And now the crack in the concrete will start to fill itself with a different mineral, and this allows you to make concrete structures that can last longer in time because they are more resilient to cracks. This also has applications in the study of biofilm formation on bone prosthetics and implants. So it is not limited to one type of research. Lastly, I would like to talk about smart active systems, where you have agents which have a set of parameters that control the way they behave, for example, the way they swim or the way they interact with other agents. And you can start a very large number of simulations. In each of them, you have these agents with a different set of random parameters. And whenever the simulation leads to a behavior that you are interested in, for example, a bacteria is able to solve a maze and exit the maze, or agents can accumulate on both ends of a rod and turn it, or objects swarm around a solid object and avoid it, you will keep this subset of simulation and record their parameters. And you can now use this pool of parameters to create a new generation of smart agents and restart the simulation. And as you keep repeating these generations, you will converge to parameters which are optimized to solve a particular task. So you are effectively bootstrapping the optimal parameters to control the behavior of your agents. So ESPResSo is very flexible, as you can see. It is made in C++, which makes it very fast, but we provide a Python interface to steer the dynamics. And we use MPI for parallelization on supercomputers. For some algorithms, we provide both a C++ version to run CPUs and a GPU version. Right now only CUDA, but we plan to include AMD GPUs in the context of MultiXScale. And this allows us to make use of these new supercomputers which have a large number of GPUs installed. The source code is free open source. We made it pretty modular so that you can very easily extend the code with new features. And when you have developed a feature which is, in our view, interesting to the community, we will invite you to contribute back to ESPResSo, either as a new module in our source code or as a third-party library, because sometimes the feature can be reused in other tools like LAMMPS or GROMACS, and it makes sense to make your feature available to these communities as well. ESPResSo always prefers a third-party library over a homegrown feature, and we already leverage Verbal and Scafacos, and we plan to add many more libraries in the course of the MultiXScale consortium. We try to be fair, so the code is easy to reuse in a different context. We always invite people to contribute their code back to us or in a third-party library. When this feature is included in ESPResSo, we have a very extensive test suite to make sure that none of the existing features get accidentally broken by the new feature. We also have human review to make sure that the code is easy to understand and to maintain. If we have any question, we'll kindly ask the contributor to please explain or clarify something, or add extra tests or add extra information in documentation. And we try to make the source code available as easily as possible. We package the code for different operating systems like Fedora. And we also made ESPResSo available on Easy. So if your supercomputer or your cluster are using Easy, you are able to use and download ESPResSo right away. And with this, I would like to thank the MultiXScale Consortium for funding this research and development, as well as all our partners that are contributing to ESPResSo, and all of you for your attention.
So thanks a lot, Jean-Noël and Rudolf, for this interesting introduction to your code and the many different application cases. So the floor is now open for questions.